Our final speaker today is Pete Thorn, who's going to be presenting on the Far Eastern Curlew Research Project. Uh, Pete joined the BirdLife Southern New South Wales Branch Committee last year in 2025, and he is an active member of our very active Conservation Subcommittee. He's also the lead on our Sydney Shorebirds Research Project in Port Hacking, and he's going to present you a quick update on that shortly. Pete is an emeritus professor at University of Sydney and currently the chair of molecular and cellular physiology. He is an accomplished and avid wildlife photographer. If you are interested, go and have a look at his website. He's got some great photos on there. And he tells me he's been a birder since he was 6, which actually beats me. So I think that's all fabulous. Pete, over to you. Share your presentation and all yours.
Okay, thanks very much then, David. So I'm going to try and share it now. Slide share. I guess you can see the slides, can you?
Yes.
From the beginning, right? Okay, fantastic. Well, thanks very much then, David. So David's given me 10 minutes, strict timing here, and so I'm going to rush through all of these slides. And I'm coming to you from Janali, which is the land of the Darawal people, which is South Sydney, which is the area that we're talking about. So just as a brief introduction, this is kind of colloquially termed the Far Eastern Curlew Project. It has a focus on port hacking, which is South Sydney, and aims to get a better understanding of the birds. We want to try and raise the awareness of the birds, particularly with the local residents in Bundina and Myanmar. And ultimately, we want to use some of this data to help lobbying for the protection of the curlews. And this project has been funded by Twitchathon, and Elizabeth Dark was the primary person that put that forward a year or so ago. And also, we've got additional donor contributions that are part of the funding. So just to give you some orientation, because I know not all of you live in South Sydney. So this is the area that we're talking about. And so many of you will know about Tara Point, which actually is a national park, and it's a Ramsar listed wetland. And there's something like 50 to 100 curlews that are present there. If you walk along here to the headland in Woolooware, then you'll occasionally see one or two curlews. But the area that we are concentrating on, and let me just get my laser pointer here, the area we're concentrating on is down here, which is the Port Hacking area and Myanmar and the Demon Spit. And there is typically around about 20 curlews. So I thought I'd show you some movies of the actual birds that we're talking about. So these are curlews taken in on the spit, and this is as they're roosting, and there's about 20 birds here. And I think over on the right-hand side, you can see there's a couple of godwits. I particularly wanted to show this movie because it shows you an example of the birds feeding, and that's going to come up next. This is typically how you would understand what the birds are feeding on. You just look at them and see. And I challenge you to understand from that picture there actually what that bird was eating. It probably was a yabby, but it's very difficult to pick that up. And I'm going to show you how we've used eDNA to actually analyze what the birds are eating. So this is the Far Eastern Curlew project. It's a 2-year project. The formal aims are firstly to define a spatial map of how the Far Eastern curlews are using this area in the Port Hacking Basin. Secondly, to understand what the eastern curlews are eating, so what their diet is, and then to map their prey distribution. And finally, to use this quantitative mapping to try and look at the disturbance factors, which include the presence of people, watercraft, and dogs. This is a very heavily used area for recreational activities. And there are two people involved in managing the project with me, and that's Chris Kelly and Chris Binns. So this is just showing you a map of the area, and you can see here, this is the spit, the so-called Deepen Spit. So at high tide, this is the area that remains, and this is where the birds will tend to be roosting. And then at low tide, as the tide goes out, then this area over here gets exposed, and that's the mudflats. And so the birds are using that as their feeding area. So the first aim is to try and understand exactly how the birds are using this area. So what we've done is we initiated a citizen science project, and we've recruited a number of volunteers. And essentially what we've done is we've mapped out this area into a grid square, as you can see here. And so what the citizen scientists are doing is at any point in time, and they could go out at any time of the day, then they would go out and say, oh yes, I can see, say, 5 birds in C5, and I can see 3 birds in B4, something like that. Now that gives us an idea of how the birds are distributing at that particular time. We know the tides in this area, and so we can relate the time of observation to the time of the tide. So let's say the observation was 11 o'clock, then we can say, oh, that's an hour before high tide, right? We can accumulate all the observations, and then that gives us an idea of how the birds are actually using this area. This project has been very successful. We have 250 observations to date. And remember, we're talking about the last summer, which has just been completed now. We've got data available across the whole day. So that means these observations are right from dawn till dusk. And they're across all the tide cycles. So that's really a fantastic data set that we've got. We're seeing an average number of about 18 birds. And overall, we had about a bit more than 10 people doing the observations. But I specifically here want to credit Julia Keating, who is a resident of Myanmar, and she's made the majority of the observations that we're using. So I don't have time. I've only got 10 minutes, so I don't have time to show you all the data, but this is a snapshot of what we've got. And here, what I'm showing you is a heat map of where the birds are distributing in the 3 hours up until high tide. So it's important to remember here that when I say 3 hours, this is an accumulation. So this is 3 hours before high tide, right up to high tide. So what you have here is a heat map of the distribution of the birds. The green means that there are low numbers of birds. The yellow means that it's relatively higher numbers. The orange is higher, and then the red is even higher still. So what we're observing here is that the birds are moving from the tidal flats across to roosting areas down here. And you can see that they have a preference for this particular roosting area, which is G7 right at the bottom there. Okay. So I've been working on this with Saif. I should have mentioned Saif earlier on. So Saif is a student from Macquarie University, and he's helping me do all this analysis. But the conclusion, or the interim conclusion that we've got here, is that the birds have a preference for this one area down here, which is G7. And so this gives you an example of how we might use this data for lobbying, because now we've identified the preferential area that the birds are using for roosting, then this is something that we can go to the local council and ask for protection of that particular area. And fencing off that area would then provide refuge for the birds where they could actually roost in that time. As I say, I don't have time to show you all the other data, but we're working on all the other tide times, and we're also working on the impact that people have on this distribution, because as well as observing the birds, the observers also were counting how many people were there, how many boats were there, and how many dogs were there. And we're looking to see how that might impact on this distribution mapping that we've got. Now, the next thing that we wanted to do was to identify the curlew diet and map out where their prey is existing. And I've just got this cartoon representation here because I think this is much more of a general application for many of the projects that BirdLife Southern New South Wales may go on to work with. And that is that we've chosen to use a technique called eDNA, which is actually an extremely powerful technique that is now being very broadly. And what you can see, well, being used very broadly, and what you can see here is that you can just take samples from anywhere in the environment and then run them through this eDNA analysis and identify, various species that might be present there. Now, in our study, we've particularly taken samples from the scats of the curlews because we want to understand what the curlews are eating. And we've also taken samples from the mudflats to understand what is actually living there. So with those samples, we've submitted them to a lab called Whittler Labs. They process them, they amplify those samples, and then they compare them to a database. And then you can get this kind of information. And so this is just showing you a whole shopping list of the kind of things that you can do. Now, specifically, what we've been doing is to use this top one here, which is species assemblage, which just means to identify how many species are present in that particular sample. But you can see you can use it for all sorts of different things. And I thought a good example for this audience would be to say how people are using it, say, to map the distributions of platypus. Because what you can do there is just take a sample of water from a river and you can immediately identify whether there's a platypus living there or not. And if you weren't using this technique, that would take you hours and hours of observation to understand whether a river had a platypus or not. However, to go back to our test, now what I'm showing you here is just an example of the data that we've got from the scats. And on the right-hand side, you can see there are 5 columns of data here, and each one represents an individual sample of an individual bird poo. Okay. Now, when you're looking down the side over here on the left-hand side, you can see the species that have been identified. And the top hit that we've got here is actually curlew. And that's what you would expect because in the curlew poo, they're pooing out many of the cells that they've got. And of course, that's got the curlew DNA in there. So that is the top hit. But what's really interesting about our data is when you look for the next hits here, you can see that these next 2 here are Tripea, which is actually the Australian ghost shrimp. And you can see that the important thing here is that that is the top hit in every one of these samples. So all 5 of the samples, and in fact, we've got another one which I'm not showing you, all 6 of the samples have top hits of a prey item, which is the ghost shrimp. And again, I think that is information that we can use. What we know about the ghost shrimp is that recreational fishermen use it as bait. So they're digging them out of the sand and they're using them as bait. And also we know in Port Hacking that there is a commercial license for fishing of the ghost shrimps. And what we can do with this data is now add the curlews there as an element that needs to be considered if we're considering about the ghost shrimp population and how to protect their numbers. It's not just the recreational fishermen, it's not just the commercial fishermen. Now we've got to factor in the impact that it might have on the curlews. So, in summary, we're halfway through the project. So, this is a 2-year project. We've achieved a quantitative map of the bird distribution across the area. And right now, Saif and I are working on how this is being impacted by disturbance by people and dogs and birds. We've achieved an initial assessment of the bird diet and prey distribution using eDNA. I've shown you the eDNA results from the bird poos, but we've also done eDNA studies of the mudflats. And in the future, we are going to continue the DNA work to build up a scientifically robust data set, looking at the bird scats. We're presenting the findings at a Shorebird conference in Newcastle in September, and we're planning to expand the project to potentially include bird banding and possibly telemetry. And that's it. I went a bit over there, David, I'm afraid.
That's all good, Pete. Look, I am very excited by this project and where it's heading. I think the potential insights and data we're getting from it are going to be fabulous in terms of informing the way we can campaign and advocate for Far Eastern curlews in Port Hacking and beyond. So I'm very excited by where it's heading. Does anybody have any questions for Pete Bales?
Thank you. Thank you, Peter. Excellent. I'm wondering if you have any historical data that you will be using to compare with data that you're building up now. And thanks for mentioning Julie, by the way, because no one knows that area better than her.
Yeah, exactly. I mean, I think she was the inspiration for the project. So I think we need to, we are a great vote of thanks. Look, I mean, Julie has made these observations over, I don't know, 10 or 15 years about the birds returning. I think what this project does is it adds a level of kind of science to that because, for example, when I show the average number of birds, that over 250 observations with a number of observers there, I think that has quite a lot of strength. I would
Yes.
also say, Bales, that the essentially when we're looking at that roost site, I think, when you go and visit that site, the birds are using those sites, right? But I think these observations really lend weight to that. It's clear that the birds are using that. And I think what will be very interesting that we've not known before is the kind of impact of the disturbance, right? So if we can get a measure of that, and I wasn't able to show any of the data because we're still working on that, but if we can get a measure of that, then that's important. And then the final thing I'd say, which I'd add to what David says, is I actually think this contributes to the scientific knowledge of the species. Nobody, to my knowledge, has done an eDNA analysis of what the birds are actually eating. And I hope you picked up there when from the movie I showed you at the beginning that prior analysis has just been observational, just looking through binoculars and saying, oh, I think that was a crab, I think that was a, whatever. And so the DNA is much more precise. So you can say, yes, I forgot to mention it, but as you go down that shopping list, you can see that they're eating worms, they're eating other kinds of crabs. So we know precisely what they're eating, and even to the level of knowing what that individual bird was eating, right? Because it's all in the individual poos. So I think that I'm hoping that that's going to add to the our scientific understanding of curlews. So.
Thank you, Pete. We've got one time for one more question. I think Jenny, you've got your hand up.
Hi, Pete. Um, oh God, sorry. Are you also sampling across the whole area to understand the whether the ghost shrimp is across the whole area, or are they going to areas where the ghost shrimps are in higher concentration?
I mean that's a good question, Jenny. So what we did is that we've sampled across the mudflats where the birds are, and we took actually 6 samples from there. We also sampled elsewhere, and we only took 1 sample. This was just us. I mean, right at the outset, we didn't even know if the DNA was going to work. So I mean, I think this is very positive that it's actually showing us amazing findings, really, right? Yeah, so that's on the cards. And I need to have a discussion with everybody about that, whether we use the eDNA to actually map out the whole of Port Hacking, right? And then just say where are the prey species existing? And previously we've argued amongst ourselves to say, okay, well then that might give us a precedent to say, well, actually, we need to protect this particular area because this is where the birds are focusing in on because the prey is there. So yeah, we're debating that. We've got the next season coming up, which will be in, September when the birds come back, and that's when we'll start this project up again. So we'll see if that's something that we end up doing.
I guess the counter argument is that if you show a map like that, then the commercial fishers know where to go to get the coast shrimps as well, and so you've got competition.
Well, I mean, it's a bit like bells. I mean, we kind of already know this, Jenny, because the pumpers, like, so not the commercial fishermen, but the recreational fishermen, they know where those things are. So when you go there, almost any time of the day, there'll be somebody pumping out yabbies, and they don't do it anywhere else in Port Hacking. So that's obviously where the yabbies are. So there's an element of kind of self-fulfillment that if we could spend thousands on a DNA survey across the whole of Port Hacking and just see what we already know. So I'm not sure. One of the things I'm very interested in is seeing if we can compare this population of birds with another population of birds elsewhere to see if these ones are specifically targeting the yabbies because they're here. And if we went elsewhere, for example, Shoalhaven Heads is the one that I've got in my mind. I'm not sure there are yabbies down there, and we could do DNA sampling down there, and then we could see what that was like. And I think that might be an interesting question, right?
I think Pete, much like Adrian said earlier, if we let you go, you'll keep talking on this for another hour. So I'm going
I
to I'm
will.
going I'm going to halt you there.
Yep.
And thank you very much, Pete. That was fascinating. I'm really looking forward to the conclusion of this project in a year plus and see where it takes us. I think it's very exciting.